Okay, so thank you, Dagmar, for the invitation, for that nice introduction. And welcome, everyone. I appreciate everyone joining a talk online. I know we spend most of our days on Zoom, and so I'm very appreciative that at the end of your day, you're choosing to be in yet another Zoom meeting. My talk today is titled "The Mechanics of Human Limbs, How They're Regulated by the Nervous System and How They Influence Neural Control." And what I've tried to do today, when Dagmar invited me, I think 2 weeks ago or so, to give a talk here, I've had the pleasure of speaking at an action club in Pennsylvania before, and so I've modeled my talk a little bit on what I experienced then, and it's meant to be very informal. I have two parts. The first part is almost tutorial-like, and it's really giving a bit of a background on what do we mean by limb mechanics, how do we measure limb mechanics, and what are the ways the nervous system can control them, and how are they traditionally thought about in the context of motor control. And that's going to be an overview of some work from my group as well as work from a number of other groups. And the last half of the talk is going to be more a detailed presentation on one of our current studies we just finished. Again, interrupt at any point, because I've actually broken it up into 2 as opposed to 3 talks. I changed that from my initial email to Dagmar to this morning when I finalized my slides. But interrupt along anyway, and I'll try to put a natural break in there as well. Okay, so focus generally in my laboratory, the questions we try to answer are really pretty simple. It's how do we regulate the multi-joint mechanics of our limbs to do the tasks that we take for granted every single day, whether you're grasping onto a soccer ball or running through mud, or if you have the skill playing an instrument, which I don't, you have to change the mechanical properties of your limbs to accomplish each and every one of these tasks. But it's something that we don't really think about. But there are many components to this ability that we take for granted. One is the mechanical properties of our limbs. What are the muscles and how do their properties change when they're activated by the nervous system? This is neurophysiology that allows us to activate muscles and change their mechanics. And then in order to study this, we need a number of engineering techniques. So my lab employs some musculoskeletal modeling. We do a lot of signal processing and system identification. And we try to take this fundamental knowledge and these tools we're developing to study problems in rehabilitation, as Dagmar mentioned. So my lab is at the Shirley Ryan Ability Lab, which you may know as a number of you trained there before, back when it was the Rehab Institute of Chicago. Today, I'm going to mostly focus on a pretty fundamental talk looking at neurophysiology and biomechanics in the context of motor control. And to give you a rough outline, so again, I'm going to start by giving a little background on some of the physiological mechanisms for regulating limb mechanics. So there are many groups that have been studying limb mechanics and how limb mechanics change across various tasks for years and years. And where my lab tries to position itself is we really try to focus on what's the physiology underlying that ability so that we can understand, excuse me, when the physiology is impaired, what are the corresponding functional impairments? Many groups that have studied limb mechanics have really focused on tasks where stability is important. So I'm going to review a few of those studies that have been done from our groups and from others. And the first half here, again, is really just to give us all a common background. Dagmar informed me that you're coming from engineering backgrounds, clinical backgrounds, and motor control backgrounds. So hopefully this puts us all in the same playing field. And then, as I mentioned before, I'm going to go into a more in-depth on a recent study that we've done showing how the nervous system can leverage limb mechanics to actually simplify the problems it has to solve. And I think this is something that's been proposed by a number of groups, and I think we have some really nice convincing evidence that this is indeed done. Okay, so as I mentioned before, the main focus of my talk is going to be on some fundamental questions in motor control and biomechanics. But I wanted to motivate some of this by some of the applications that we and many others have done. So why does this work? Why are we interested in understanding the mechanics? In addition to understanding changes following neuropathologies, there are also many folks who are building artificial devices to restore the ability to walk. A former student who worked partly in my group and partly in others at the Rehab Institute is Elliot Rouse, who did some really nice studies measuring the mechanics of the ankle during locomotion. And his motivation for doing this, and something he continues to do in his own laboratory, is to build artificial devices that match the mechanical properties of the human limb. And this is a video I like. It's an old video, but it's one that when he first took his experimental measurements that he took on the human ankle during locomotion and programmed them into an artificial prosthesis, what you're going to see here is someone walking with this device the very first time. And I want you to just notice how natural this is. We were amazed at how well this worked. Now the subject's going to go toe off. This has nothing to do with the impedance measurements. And you just watch again when it comes back. So this is just one example of how a knowledge of limb mechanics and how they're regulated in functional tasks can be used in the rehabilitation process. We have two other main projects we're doing in my group now, which I'd be glad to talk to you about, but they're not in my presentation, but I think are really interesting. One looking at shoulder mechanics and shoulder stability and how changes in neural control can lead to a number of different pathologies, including osteoarthritis and multidirectional instability. In a more recent study, which is looking at changes in motor control associated with chemotherapy, particularly on the sensory side of things. So again, all these fundamental studies we do feed into these clinical applications. Okay, so if we're interested in limb mechanics, the first thing we need is some way to measure them or quantify them. A technique we use a lot is we measure the impedance of joints or limbs. And so in general, the impedance of a limb describes the resistance to external displacements of posture. Now, impedance is something that's not fixed, right? If you look at these soccer players juggling a ball, you can see the many different ways they control the impedance of the limb. When the ball comes to them and they trap it on the knee, the leg has to be compliant, so the ball doesn't just bounce off. On the other hand, when they bounce it to their foot and pass it, then you need a stiff ankle in order to have a rigid surface, and you can impart both force and energy onto the ball. Now, in many situations, a number of groups have shown that the impedance of joints or limbs or muscles can be modeled often by a system when you consider its mass or inertial properties, its viscous properties or damping, and its elastic properties or stiffness. Today, I'm going to mostly focus on stiffness because over the many tasks that we do, which tend to operate at a relatively low frequency, such as locomotion or postural tasks, stiffness tends to be the mechanical property that dominates, and I'll show some evidence of that later on. But much of what I'm going to focus on today is talking about the stiffness of muscles, joints, and whole limbs. I also want to just talk a little bit about how we measure this. The basic premise of how you measure the mechanical properties of the limb is you have to perturb the limb. You measure by a set distance, you measure the forces generated in response, and you map out the relationships between those displacement perturbations and the corresponding forces. This figure here is a technique I used in some early work back when I was a PhD student, but it was really pioneered by a lot of work that Neville Hogan and Sandro Musavaldi did across the river for many of you who happen to be at Northeastern right now. And the basic idea here is if you wanted to measure the mechanical properties, specifically the stiffness of the arm, what you could do is take a robotic system, perturb the arm, whoops, you can see my cursor here, perturb the arm by a set amount, measure the forces generated in response. So here we've got time traces on the bottom, spatial traces on the top, and you can characterize the relationship between these unit displacements and the corresponding forces to get a characteristic endpoint stiffness of the limb. And you can represent this graphically by essentially just connecting the dots. And so what you can think of this in multiple dimensions is it's a multidimensional spring. If you did this on a single muscle, you'd be measuring spring-like properties of that muscle. Now, there are many physiological mechanisms that contribute to the impedance, or in the case of slower movements, the stiffness of our limbs. Obviously, the geometry of the limb, the posture, muscles and their activation, the structure of those muscles, and then of course, how the nervous system activates them. And so today I want to spend a little bit of time focusing on these mechanisms in my next few slides and how they can contribute to the mechanical properties of muscles and limbs. Now, there have been many seminal works on whole muscle mechanics, and many of my favorite papers were done by Rack and Westbury back in the late 60s and early 70s. This is a paper where they specifically looked at a muscle property known as short-range stiffness. And the basic idea here is that when you stretch a muscle rapidly, it behaves in a very spring-like fashion. Now, if you have done much muscle modeling before or musculoskeletal modeling, there's a property of muscles that is in almost every model known as the length tension curve. And that's the dashed trace. I'm pointing, I think, to the wrong part of my screen. This is a dashed trace you can see on these this slide here. You'll see that there's a passive condition down here. So the passive properties of muscle, if you were to stretch a muscle, so increase its length, its force would increase. And then as you change length and then change activation, you'll get different forces as well. What I want you to note though is that while most musculoskeletal models just consider this length tension property of muscle, what really matters in the case of at least postural control and interacting with the environment is if you stretch a muscle at any given point along this curve, the force it generates increases dramatically. This increase in force is known as the short range stiffness. Now, short range stiffness has been known as an important property of muscle. It's the muscle's first response to an external disturbance before the nervous system can ever intervene. What has been unknown until a little while ago is how relevant this property is to the mechanics of entire limbs. So quite a few years ago now, we set out to do a number of animal studies to see if we could characterize. Yeah, John, go ahead.
Hi. Is this short range stiffness, is this like monosynaptic stretch reflex, H reflex stuff, or is this something different from that?
Yeah, great question. No, so this is simply an intrinsic property of muscle. So in these measurements here and in the ones I'm about to show, the reflexes were disconnected through by disconnecting the afferent pathways. So this is purely a mechanical property, thought originally to be largely dependent on the number of cross bridges that are active in a muscle. It's a bit of a controversial topic these days, and a lot of people suggest that it's probably a combination of the attached cross bridges as well as the extracellular matrix.
But there's no neurons involved in this.
No, other than that, we're activating each measure. Muscle has a different short range stiffness depending on how active the muscle is. Essentially, it scales with force, but no feedback. Great. Okay, so one of the questions we wanted to do in order to see if we could understand how short range stiffness was contributed to the mechanical properties of whole limbs is we first needed a way to scale these results from animals where you can measure short range stiffness to humans. So we did a number of studies in the cat hind limb where we dissected the distal end of a number of different muscles, connected the distal end of the muscle to a muscle puller so we could make direct mechanical measurements over a range of different activations. And what we found was that a very simple model of muscle allowed us to characterize short range stiffness. And we only needed to know two things about this muscle. We needed to know, and they were both geometrical parameters. We needed to know its cross-sectional area, physiological cross-sectional area, so we could predict its maximum force. And then we also needed to know the geometry of the tendons, the length and the cross-sectional area of those, because we had a material property for tendon and we had a force-dependent property for the short range stiffness. And what this slide is showing is it's comparing our modeled results, which are shown by the solid lines and the confidence intervals, with experimental data, which are the circles here. And so these models were not fit to experimental data. The models are generated based only on the geometric properties of the muscles we measured. And you can see that the experimental data fits those models extremely well. So this is nice. This means that we have a scalable muscle model for this one property known as short range stiffness. And then
just more on the geometric properties of the muscle, because and maybe I just got distracted here. It's not a muscle model based on the cross bridges. It's on just macroscopic features and generic.
Yeah, no, that's right. So there's basically, we take, so we have two parameters that we've identified through experiments. There are a number of experiments on muscle. One parameter is essentially the Young's modulus for tendon. So it's a material property of tendon. And then we can get the stiffness of the entire tendon simply by scaling the Young's modulus by the geometry. And we have a similar material property for muscle, which is the Young's modulus for characterizing short range stiffness. The difference is that the short range stiffness Young's modulus is force dependent. So it changes with the activation of the muscle. Okay.
Okay, thanks.
So then the question is, so this simple model worked well in the cat, but how well does it scale to humans? So what we then did is we took our muscle model generated from cat data, we scaled it up to the human arm, we plugged these into a musculoskeletal model that my colleague Wendy Murray developed, and then we tried to compare it to experimental data. So here is some of the experimental data that we used. These are old data, again, that I collected in my PhD, and then we modeled these based on the cat data more recently. And let me just walk you through these experimental data first. So what you're seeing here is the experimental measurements of endpoint stiffness from a human subject. Let's look in the middle here where you'll note the difference here across the three sets of data is the posture of the arm. So in the middle case here, the hand is positioned approximately in front of the shoulder. Here. And then each of these endpoint stiffness ellipses is located at a position proportional to how hard the subject was generating force. So this endpoint stiffness here at the right, the subject was pushing rightward with a force of 50 newtons. This one up here, they're pushing outward with a force of 50 newtons. So as you can all imagine, as you increase muscle activity, your limb becomes more stiff, and that's all that you're seeing here. So as the subject generated more force, we had more endpoint stiffness. You'll notice another important feature of endpoint stiffness, again, this was shown long before us by Sandra Musavaldi and Neville, was that as you change the posture of the arm, the endpoint stiffness also changes. It can change a bit in magnitude, but mostly it changes in direction. So what we did then is we took our muscle models and we simply estimated the activation of the muscles in the arm. We did a simple optimization so as to estimate the muscles, the activation of each muscle is based on the net force the subject was generating. Okay, so we didn't actually fit these experimental data. We just plugged in a force for each of the muscles in the arm based on that optimization criterion. And we compared our predictions to the experimental data, and you could see that they were fit remarkably well. And so what this suggests is that this property of short range stiffness is actually incredibly important for understanding the mechanical properties of the limb, at least under the experimental conditions that we used.
Do you think you would get the exact same results if you change? So at the moment, so let me understand, at the moment, you always are at the same joint configuration. You are just increasing the force in the directions. Or are you actually moving to the center of those ellipses and measure the stiffness ellipse based on this displaced endpoint?
Yeah, good question. So each of these sets of endpoint stiffnesses was measured at a single posture. There are three different sets, each set at a different posture. So in this case on the left, the hand is positioned more medially, about in front of the trunk. Sorry, there's a spelling error there. And over here, the hand is positioned more laterally out to the side. So there are three different postures here. At each posture, we looked at multiple different muscle activations or forces. And so you ask a really interesting question. And so these results are all done at a given posture. If we were to move from one posture to another, this model actually fails. It fails pretty dramatically. And so there are a lot of experiments we've been doing lately looking at characterizing changes in limb mechanics during movement. And a big thing that you notice is that the predictions you would make from postural measurements are off by about a factor of 2.
Okay.
I have another question on. So the blue ellipses are the short range stiffness model, but if I recall from the last slide or whatever, there's a lot going into that model other than just that short range stiffness component to it. Do you have any sense of, I don't even really know if this is a well-formed question, but like, do you have a sense of like how much of the goodness of this fit between the model and the empirical data is because of the short range stiffness versus like other aspects of the model being good?
Yeah, so there are really only two components of this, and I'm going to explain those in my next slide. The two components are a short range stiffness component. So basically an activation dependent change in muscle stiffness, or really the change in stiffness of those muscle fibers. Okay, and the second component is a activation independent material property of the muscle. Sorry, excuse me, I said that wrong, of the tendon, material property of the tendon. And so that's it. So in this case, it's only those two properties that could contribute to these mechanics. In the arm, most of the tendons of these muscles are relatively small. So I think what we're seeing here is largely a property of the change in muscle stiffness with activation. But that's a perfect segue to my next slide.
Sorry, yes. I don't have the hand function on my Zoom.
Okay.
In your previous slide, so I suppose there was no kind of co-contraction or any kind of subject stiffening up, and you don't have similar effect in your simulations.
Yeah, that's correct. So we had, when we analyzed the experimental data a long time ago, that was one of the things we found. In these particular cases, there was very little co-contraction. If there was, and so the simple optimization we used to estimate how active each of the muscle was worked just fine. In a more complicated case with lots of co-contraction, this particular approach wouldn't work. But however, if you did know, if it was a postural task and you did know the activation of muscles, so we've done some work at the knee where we purposely had subjects co-contract. And if you measure EMGs as a way to estimate co-contraction, then the same model works. But again, it works for posture, it doesn't work well for movement, and I'll show you some data for that later. Okay, so back to John's question. So there are two components to this model, right? And there's essentially a muscle component and there's a tendon component. And so we know that there are many physiological conditions, the simplest one being aging, where mechanical properties of muscles and tendons change, right? So the scaling model we worked, the scaling model we developed, or scalable model we developed, is unlikely to work. So we've been looking at a couple different ways to try to directly measure muscle and tendon stiffness in human subjects. And I'm going to show you one of those, which uses a similar method to the one I've talked about already. And this is really quite simple. So we've been using ultrasound to measure motions of the muscle tendon junction. So if we were looking at, I'm going to show you some data if we're measuring the mechanical properties of the ankle. So we apply a displacement to the ankle, very similar to the displacement to the arm I was showing you earlier for measuring arm stiffness. And you can imagine that to a first approximation, the ankle is connected to the tibia through a muscle that has a tendon and has muscle fibers. So if we were to measure the muscle tendon junction, then we should theoretically be able to disentangle the contributions from tendon or the tendon mechanics from the contributions from muscle fibers. And so we've developed an experimental paradigm that lets us do that. We apply rotations to the ankle, the pseudorandom rotations. We use B-mode ultrasound to image the muscle tendon junction. And then from these two measurements, let's put all these up, we can estimate two things. So from the ankle motions and the ankle torques, we can measure ankle impedance. And this has been done since the late 60s that people have been making these kind of measurements. The thing we've added now is from these displacements and from our measures of the muscle tendon junction, we can also estimate a quantity we're calling the impedance ratio, which is simply a ratio of it tells you how much of the motion of the ankle gets transmitted to the muscle tendon junction. It's also the ratio of the impedances of the tendon to the entire muscle tendon unit. And there are a couple of things I want you to notice here. So I told you earlier that while the general mechanical properties of a muscle tendon or joint or whole limb are characterized by a general impedance, for at least lower frequency components, it's the stiffness that really matters. So if you look below at about, this is 1 hertz to about 3 hertz, the mechanical properties of the ankle are largely flat. So this is a frequency response function. This flatness here indicates that the stiffness is what dominates. And this is even more true if you look at this impedance ratio, which is extremely constant in this range. So I'm going to show you results for the stiffness component of, I'm going to use these two quantities that we estimated, the ankle impedance and the impedance ratio, and with a little bit of algebra, we can get the stiffness of the tendon and the stiffness of the muscle fibers.
Sorry, this impedance ratio, I'm still mulling over. So firstly, it's below 10 to the 0. Where is it? And what does it mean? What is higher and what is lower?
Yeah, good question. So the easiest way to think about this is how much of the motion of the ankle gets transmitted to the muscle tendon junction. Okay, and you could imagine that if your tendon was infinitely stiff, let's say it's just a wire or a rod, then all of that motion of the ankle is going to get transmitted to the muscle tendon junction.
Mm-hmm. Yeah.
Okay, and so in that case, the impedance ratio is a ratio of the tendon impedance to the entire impedance of the muscle tendon junction. So if the tendon is really stiff relative to the muscle, the impedance ratio is 1.
Mm-hmm. Yeah.
If the tendon is very compliant relative to the muscle, the impedance ratio goes to 0.
Okay. Okay.
Okay, so with these two quantities, we can disentangle muscle stiffness from tendon stiffness, muscle fiber stiffness from tendon stiffness. So let's first look at muscle stiffness, and these are just some sample results from 4 subjects. As you'd expect, as muscle force increases, okay, so we estimated muscle force from the ankle torque and we scaled by the moment arm. As muscle force increases, stiffness increases, as I showed previously and lots of folks have shown. How does this compare to those cat data that I showed you a little while ago? Well, if we take our cat measurements, which was the short range stiffness parameter that scales with muscle activation, and plot that here, again, this is not a fit to these data. This is just plotting the results from the cat. You can see that we see very similar scaling in humans. This is pretty cool. This can, again, matches what we saw in the arm. Let's look at the tendon properties. So you'll notice here the tendon properties, you can see I plotted these on the same scale. You'll actually see that the tendon stiffness increases slightly with force also, but at a much lower slope than the muscle itself or the muscle fibers. These results also compare quite well to cadaveric studies that have been published before. It's very hard to find any in vivo studies in humans, or at least we haven't found any good ones that cover the entire range that we can estimate here. But these cadaveric studies, particularly at higher force levels, work pretty well. We're estimating much lower stiffness at lower force levels. But so this gave us some confidence in our measurement. So let's just look at all the data we've collected. This is about 12 subjects so far. And this is just the results of our model. And there's something really interesting, I think, that's really important here. If you look at the mechanics of the ankle, you'll notice that the muscle stiffness or muscle fiber stiffness is shown here in red, and the tendon stiffness is shown here in black. You'll notice that the tendon stiffness is much, much lower, almost an order of magnitude lower than the muscle at high forces. So what does this mean in terms of mechanical properties of the ankle? They're dominated by the tendon, because you're always going to be dominated by the most compliant element, right? So even though we can change the stiffness of the ankle, we can't change it anywhere near as much as you'd expect based on the mechanical properties of muscle alone. So this is a student who's continuing to go on, and what she's really interested in is looking at changes with aging. And so we think she has a nice way to really disentangle changes in muscle stiffness and tendon stiffness with aging.
But let me just quickly react to that. Isn't that, I mean, counterintuitive that muscle stiffness is actually higher than the tendon stiffness?
Yeah, so it's going to depend. It's going to be very different depending on which muscle you're looking at. So this is the Achilles tendon, which I believe is the longest tendon in the human body. So it's really long and thin. And so it tends to be quite compliant, except for at the very lowest contraction levels. If you were to look at a muscle like some of your deltoid muscles, which have very short tendons compared to the muscle fibers, then the muscle fibers are going to dominate. So this gets to the point that we have to really consider the geometry of the muscles and the tendons in order to understand the full properties.
Mm-hmm. Mm-hmm.
Neville?
Just was curious now, do you have enough information about the geometry of the variation of moment arm about the joint? And is the increase of stiffness of the tendon sufficient to stabilize the limb? So for example, if the tendon was to have constant stiffness, as you increase force, it becomes less stable and ultimately unstable.
Right. That's a good question. So we've looked at a sensitivity analysis on moment arms, so there's not great data. So we can get a reasonable estimate, but we certainly don't know how the moment arm changes over the entire range of motion. What it seems to, and I haven't done the stability analysis, actually, that's an interesting question. So there have been a lot of studies in particular with respect to standing. I'm sure you know some of the work in Morasso et al. That suggests that stability in standing is actually an active process as opposed to just a passive property of the joint. So this would suggest that he was probably even more right than he was thinking. So, but that's an interesting thing to look at. Yes.
Excuse me. Hi. I wanted to wonder for Achilles tendon, how important is the stiffness as reaction to the torque compared to stiffness for stretching? Physiologically, how relevant is this stiffness at all?
Yeah, so this stiffness is a stiffness that results, so it's a change in force for a change in length, so the stretching that you're talking about. The torque component here is that stiffness changes with torque. So for example, if I look at my elbow and I relax all my muscles and I try to do a stretch, I'm not very stiff. But now if I increase the torque, I'm all of a sudden very, very stiff. So in all cases, this is a stiffness or a change in force with change in length.
So it is the torque for the joint, not for the tendon.
So in this, it would
along
be,
the axis.
it's both, right? Because the force of the muscle is transmitted through the tendon. So we're assuming here that whatever force is in the muscle is in the tendon, and then those two create a torque about the joint based on the moment arm.
Thank you.
Okay, I think there was one more.
Yeah, Joe, didn't you have your hand up, or did you just practice?
No, no. My question is, this also is counterintuitive to me, because if you have this floppy tendon, you don't have good control. You would expect that you would want a very stiff tendon so that everything could be controlled with the muscle force.
Yeah, so good question, right? So the tendon here still has a substantial stiffness, so it's not like this is completely floppy. One of the things that's interesting is we had some results. In fact, they're the results that went into that prosthetic device that I showed you, where we measured the ankle impedance during locomotion, or the ankle stiffness specifically during locomotion. And one of the things that we found that I made poor Elliot go back and do his analysis many times because I didn't believe the results. Because what he was finding is that the stiffness he measured was the same as what folks call the quasi-stiffness, which is the slope of the moment angle curve, which doesn't make any sense if muscle contractions change. So essentially what this allows is, because the tendon dominates, it allows the ankle to behave passively. So it doesn't have a stiffness that changes dramatically with muscle activation. And so that fits with a lot of the balance literature too, where they suggest that the way that we maintain balance is not at the ankle. This is again going to be different for different joints, but is not by regulating the impedance of the ankle as much as it is by changing torques or active control through feedback. So there's clearly enough stiffness to transmit forces and create loads, torques about the joint. You just don't change the mechanical properties as much as you could if you had much shorter tendons. Does that answer your question?
Yes, thank you.
Okay.
I think Neville had a question just earlier.
Let me hold it for later. Let me not deflect things at this point.
I have a question.
Sure. Yeah.
And that is, in terms of the tendon, Prosky and Gandevia have talked a lot about sort of the stretch of the tendon and the change in length, and taking a relatively long time for the viscoelastic restoration to rest level return. And how does all of that relate in terms of postural control, especially if people do these standing on tilted platforms and things of that sort, in terms of affecting the response properties of the muscles?
Yeah, so at least in our hands, so certainly if you take a tendon and you stretch it and you leave there for a long time, there you see a lot of creep over time, but that's relatively slow. So if you look at this figure here, the blue trace on the bottom is essentially a measure of tendon properties. And so we've got right, so this is a dynamic curve. You can see down to about, this should be about a half a hertz or so at the bottom. We're still seeing no change in tendon properties. So if there's creep, it's much slower than at least a half a hertz. So it should be something that the nervous system can compensate for.
All of their stuff involves a relatively long time period. So you're out of that range here.
That's right.
Yeah. Okay.
So I think over those time periods, it's probably not too hard for us to compensate for.
Yeah. Okay. Thank you.
Yeah. Okay. So I'm going to go through the next couple slides a little quickly. So just one other mechanism that I wanted to talk about is there are many different ways to control activation of a muscle. So the key point so far is we can control impedance of a limb through changes in posture, through changes in muscle activation. And we can change muscle activation in two different ways. We can do it by thinking about it, the co-contraction I did earlier, or it can happen involuntarily, right? And so probably the most widely studied involuntary way to change muscle activation is through the spinal stretch reflex, which someone mentioned earlier. And this is a classic figure from Nichols and Houck back in 1976, who looked at the force generated when stretching the cat soleus muscle under two conditions. The top curve here and the bottommost curve are when a soleus muscle is stretched or released and the reflexes are intact. And the inner curves show the dramatic drop in force and therefore the dramatic drop in stiffness when the reflexes are not intact. So this is yet another way to change the mechanical properties of a muscle. About 10 or so years later, Sinkower and colleagues showed that using electrical stimulation to block the stretch reflex, that these involuntary reflex pathways can contribute substantially to the mechanical properties of a joint. They tend to be larger in the lower limb than the upper limb, but about the ankle where these data were collected, the reflex stiffness or involuntary change in muscle activation can contribute as much as about 50% of the net mechanical properties of the joint. And then the last mechanism I want to talk to is, the involuntary pathways become much more complicated than just the spinal stretch reflexes. There are also many involuntary feedback pathways that go through the brainstem, through the motor cortex. And those are interesting because a lot of groups, including our own, have shown that those can be changed on a moment by moment basis, kind of providing an automatic gain control for these involuntary responses.
So you just mentioned in passing that upper and lower limb have different contributions of stretch reflex. Can you just elaborate a little bit?
Yeah, so this is interesting. So we have done a number of studies, mostly in the ankle, some in the knee, and then a whole bunch in the arm. And we tend to find much more robust reflex responses in the lower limb, probably because of the fact that a lot of locomotion, postural control there is quite involuntary compared to what we use our arms for. So I don't have a strong reason as to why that is, but that's an empirical finding.
And is it, well, significant as you say so, but is it by orders of magnitude, double, or what, in sort of how different?
Yeah, so when we've estimated reflex contributions to stiffness using similar protocols in the upper limb versus lower limb, it's on the order of if you look at mechanical contributions, it would be half to a third, somewhere along those lines. It's a broad rule of thumb.
Okay, thanks.
Okay, so as I mentioned earlier, many groups have, usually when people think about mechanical properties of limbs, one of the first things that they're studied in the context of is stability. So as has come up already in our discussions many times, a number of you express surprise at the fact that how can we maintain stability in standing with a tendon that's relatively compliant compared to muscles, because we can't so easily change the mechanical properties of the muscles. So let's look at some of the ways that we can increase stability through the mechanisms that I've already talked about. And I'm going to just bring out three different mechanisms. You'll notice a theme here for the first two is that there's tends to be there's a history in this field of looking at limb mechanics and stability where Neville Hogan suggests something should be possible, and it takes about 10 years for the rest of us to catch up and show that indeed it is possible. And so here's one example here is a really nice study looking at the influence of posture on the mechanical properties of the arm that Denis Rancourt and Neville did back in 2001. And about 9 years later, Randy Trumbauer, who's I think in the group was doing a postdoc with me at the time, created a task where we asked subjects to simply stabilize the hand. And they were stabilizing the hand as they were doing a particular task. They were interacting with a robot that could move in 3 degrees of freedom. And the robot was unstable. So if you held the hand precisely at the target, the robot would apply no forces. But if you moved a little bit away in one direction, the robot would push you with a force that was proportional to your displacement. So it was an unstable spring in one of 3 different directions. I'm showing two directions here. In one case, the robot was unstable in a vertical direction. In the other case, it was unstable in a horizontal directions. And we simply asked the subjects to complete this task. They were given no other instructions. And what we found is they selected postures that maximize the stiffness of the arm in the direction of the instability. So you can see when the robot was unstable in the vertical direction, they pulled the elbow down. And so we measured stiffness of that posture, and that posture tended to rotate the direction of maximal stiffness towards the instability. When it was unstable in the horizontal direction, they pulled the elbow up. And again, that changed the orientation of arm stiffness. So suggesting that when one solution is to change the posture of the arm, that seems to be the first thing that individuals go to. Another suggestion by Neville was that, maybe that one of the ways the nervous system leverages the redundancy in our musculoskeletal system is to change muscle contraction in a way that optimizes the mechanics of a limb for a given task. Etienne Bourdet did a really nice study back in 2001 where he trained subjects to move from a start location to a target location. Again, they were holding on to a manipulandum. And first they did this, they just practiced these reaches over and over, and they measured the stiffness of the limb, and this was the baseline condition. They did it again as subjects moved through a divergent force field. This is very similar to the unstable field I just described. If the hand deviated a little bit to the right, the robot pushed the subject to the right with a force proportional to how far they were. The further away they were from the straight line between start and target, the larger the force was. If they deviated to the left, the robot would push them to the left. And what Etienne and colleagues found is that the stiffness of the arm changed from the nominal condition, which is shown here in green, where they were moving just without any force field, to expand in the direction of that unstable environment. Again, suggesting that the nervous system can change mechanics to help you perform a task in a more advantageous way. In the first case that I showed, Randy's work, this was done by changes in posture. In this case, the work by Etienne and colleagues, this was done through changes in muscle activation. And one more example, this is again a similar paradigm where we looked at subjects interacting with unstable fields. And now we wanted to start to look at, well, how does muscle activation change? Does it change voluntarily, or are there changes in those involuntary reflex pathways? And so again, we had subjects interact with two fields. There's an unstable field that's color-coded in blue here, that's unstable from the hand oriented in the direction towards the shoulder approximately, and another field that's shown in red, which is unstable in a different direction. And the question is, can the nervous system understand these fields and change not only the voluntary responses, but the involuntary responses appropriately? And let's take a look here. So we're going to look at this result held in a number of muscles in the arm, but I'm going to show you some results in the pectoralis muscle, which are nice and clear. So on the bottom axis, you're seeing the changes in muscle activity in response to a rapid change in arm posture. So subjects were holding on to the robot, which created this unstable field, and occasionally the robot would unexpectedly perturb the limb. First in a direction which is to a lateral direction here, which is in the direction of this red destabilizing force field.
Under the instruction to maintain the same postural configuration.
That's right. In all these cases, they're just maintaining posture. And what you'll see is that now we look at the reflex responses that were elicited, and when perturbing in this direction, we receive the pectoralis response, involuntary response. This happens at about 50 milliseconds, much faster than you can react voluntarily, and that the response in the pectoralis was much larger when the subject was interacting with a field that was unstable in the direction of this perturbation. Now, one possible explanation is that they just, when interacting with the red field, they just simply increased the sensitivity of all their stretch reflexes, but that wasn't the case. When we perturbed, in an orthogonal direction, subject is interacting with the same two fields, now the sensitivity changes. So when we perturb in an orthogonal direction, the much bigger reflex is seen when perturbing again in the line of the instability. So I think this is pretty cool. This suggests that the nervous system is smart enough not only to change the gains of your involuntary reflex pathways, but it does it in a way that compensates for the mechanical properties of the environment with which you're interacting. Okay, so that's the end of my overview of kind of the mechanisms contributing to the mechanical properties of the limb and how those mechanisms can be used to stabilize the limb under a variety of different conditions. So just to summarize, what I've hopefully been able to show is that limb impedance arises from the mechanical properties of muscles, tendons, along with the geometry of the limb. The central nervous system can regulate impedance through changes in posture or changes in muscle activation. And those changes in muscle activation can occur either voluntarily or involuntarily. And both most likely happen in most tasks that we do. So I was going to pause there, stop my share.
So I have a question on the back to the sort of upper versus lower limb stuff.
Sure.
So like in the lower limbs, there's spinal reflex gating that happens for all sorts of reasons, not the least of which being like stance to swings transition, where your stretch reflex kind of just turns off during your swing phase and then turns on during your stance phase. It's driven by central pattern generators and all that. And I'm wondering, and so now you're talking about like, your central nervous system is tuning the stretch reflex for this various tasks. And I'm wondering, do you have any thoughts on differences in like the mechanism of how that might happen? Because I'm sure you can get a similar effect in the lower limb, but I'm just wondering if like the actual mechanism by which the descending pathways change these low-level reflexes is the same or different, or cortical spinal, or whatever that means.
Yeah, so the short answer is no, but I think they probably, in terms of do I know, but I think they're probably different. So there are a lot of mechanisms by which you can vary reflex gain. Presynaptic inhibition and presynaptic reciprocal inhibition is probably one of the ways it happens, I'm guessing, during the locomotor cycle. Certainly during movements of the upper limb, we have good evidence that that's what happens. So you'll see as you move your arm back and forth, you'll see a modulation of reflex sensitivity that's quite separate from the voluntary drive to the muscle. It seems to be linked to reciprocal inhibition from the antagonistic muscle. And I'm guessing you see a lot of that in the locomotor cycle also.
And so you see that reciprocal inhibition of the antagonistic muscle, which is a new phrase that I enjoy. That's a different thing from like the central pattern generator, or whatever is going on with locomotion.
Well, I think the central pattern generator, I'm not a locomotor person, so someone feel free to correct me if I'm wrong, but a lot of the central pattern generator, right, is going to control the amount of reciprocal inhibition. And so another reason why I think these mechanisms are different, which I should have pointed out. So I had one slide that I went through quite quickly showing two different components of a reflex response. There are more than that, but at the simplest level, there's a spinal stretch reflex and then something that comes later. In the upper limb, a number of groups have shown that much of what comes later is mediated by the motor cortex. And the kind of modulations that I showed where you're interacting with different environments, we've been able to show that that is mediated through changes in what are known as long latency stretch reflexes that go through the motor cortex, which are very different than the spinal mechanisms that I think contribute mostly to locomotion.
Is that related to the corticospinal tract, which is another phrase I enjoy but don't really understand?
It eventually comes back down through the corticospinal tract, yeah. Elliot, I think you had a question. Yeah.
Yeah, I think you answered it. I missed the latency on those adaptive responses. So they're long latency reflexes.
Yes. Yeah, so the responses that we see where there's a change based on the mechanical properties of the environment occurs beginning at in the upper limb at about 50 milliseconds after perturbation. So yeah, those we've been able to wipe those out. So the way we've done that is we've used transcranial magnetic stimulation to silence the motor cortex, and that modulation disappears.
Thank you.
Yeah.
Alice, I think you can help John on the corticospinal tract. Yeah.
But, actually, I wanted to ask, I thought it was really interesting how the reflexes can be gained in the different force fields. I was wondering how quickly this changes in gain occur. I guess it must take the person some time to figure out they're in a different force field. And if you tell them, okay, now we're going to switch the force field, can they adjust right away? Or is it sort of like something that needs to change gradually?
Yeah, that's a great question. We've tried to measure it, and it happens fast enough that we haven't been able to measure it. So the way we did the studies that I showed is we had subjects, grab them and manipulate them, move for a few seconds, and then we would take a measurement. We've tried cases where, so we have methods where we can track changes in either reflexes or mechanics over time. And so we've tried to do that where we change the mechanical properties of the force field unbeknownst to the subject. And it's really hard to measure because unless they know there's a change, we can't measure anything. So I don't have a good answer to that, but it happens within a span of a few seconds.
Oh, wow. Yeah. Really neat.
Unlike a spinal, changes in spinal reflexes, that are based on the task tend to take months of training. So there were some really elegant studies showing changes in spinal reflex, for example, in ballet dancers, but those take long periods of time.
So I think we're ready for your second round.
Okay, great. This will be a little quicker, so maybe.
It's okay.
Let me share. So, and am I remembering correctly, trying to end close to 5:30, correct?
It's announced to 6:30 sometime.
Oh, yeah, 6:30 for you, 5:30 for me.
Yeah, right. So just take your time.
Okay. Okay, can you see my slides again?
Nope.
No.
No, let me just stop.
Yes.
Yes, good.
All right, except I can't see them. Try one more time here. Okay, now you can see it large.
Yes.
Okay, so I ended by talking about stability and the role of limb mechanics in maintaining limb stability, but stability is not the primary objective in all tasks, right? In many cases, we are more interested with exerting precise forces on the environment, of throwing a friend into the air or landing safely after being thrown in the air. And a question we've been asking recently is, can the intrinsic mechanics of our limb be used to facilitate these interactions where we're more interested in the forces we exert on the environment? This is not a new question. It's one, again, that people have been looking at for a long time, but it's really challenging to answer because it's hard to dissociate the contributions from the mechanical properties to how the nervous system is changing the activation of muscles to generate forces. So I'm going to end with looking at this study, trying to answer this question. Can the intrinsic mechanics of our limbs simplify the neural control of physical interactions when stability is not the primary concern? As an example of how this is done before, one of the ways that people have tried to understand how mechanics simplify neural control is looking at motion force relationships. So this is a nice study that Dan Ferris and Claire Farley did a long time ago, where they quantified the mechanics of the body simply by looking at the relationship between the motion of the center of mass and the forces exerted on the environment. And this is a study where they're looking at human hopping. And what they did is they quantified leg stiffness as the change in displacement for the change in force. So there are a lot of reasons why this might not be a good measure of stiffness, but I think they had an interesting finding that showed that at least this measure of stiffness changed quite a bit with context. So the context of their experiment was they had subjects hop on surfaces that had different stiffnesses. And what they found that is when subjects hopped on a surface with very low stiffness, the stiffness of the leg by their measurement was high. And when we stopped, hopped on a surface with high stiffness, the stiffness of the leg dropped, suggesting that they're trying to regulate the transmission of forces from the ground to the center of mass. Here's another nice study. This one was done by Angie Biewiner's group at Harvard, where they were asking very similar question, except they asked it in goats as opposed to humans. And they looked at goats walking across different surfaces. And again, they looked at motion force profiles. Here was a motion torque profile of the hip, the knee, the ankle, and the MTP joints. And they measured, again, they looked at the slope of these motion versus, so this is a change in joint angle versus a change in joint moment. And they looked at various slopes here and tried to get an estimate, again, of stiffness. And they did this as goats walked across different surfaces, either rigid, rubber, or foam. And interestingly, they found the exact opposite result from Ferris et al. There are lots of reasons why this may happen, and I don't want to go into that. But the point here is that people have been trying to understand how mechanical properties change in more functional tasks by looking at these relationships between motion, force, or joint angle and joint torque. Now, I'm sure many of you are going, well, wait a minute, that's not a good way to characterize the mechanical properties of a limb, right? So we've talked about the fact that muscles are indeed spring-like. And if springs, if you wanted to characterize the mechanical properties of a spring, you could look at the change in length versus change in force, and that would indeed allow you to characterize the spring-like properties. But there's a problem here, and that's that muscles are controlled by the nervous system, which is kind of analogous to having a spring attached to a motor. So if I were to make a measurement of the force at the endpoint of the spring and the change in length here, but there's some motor changing the length of the spring on the other side, I wouldn't be able to independently know the spring constant. Just like I wouldn't be able to know the mechanical properties of a muscle if the nervous system is changing activation just by looking at motion force profiles. Instead, we need to know both sides of the system. We need to know what the nervous system is doing, and we need to know the mechanical properties. So the first thing we need in order to answer the question as to can the mechanics simplify neural control is we need a way to disentangle these two things. The second thing we need to understand is which tasks would the mechanical properties of a limb likely be beneficial. And there have been a lot of really nice animal studies that have tried to tackle this question. This is another earlier study from Andy Bienwinter's group looking at wallabies hopping. And again, they're looking at the length force relationship, length force relationship in two different muscles, plantaris and lateral gastrocnemius. And you can see particularly in the plantaris muscle, there's a large range here where there's very little, a strongly linear relationship between the force and the change in length, suggesting that the muscle behaves quite spring-like. And they were able to show that the animal leverages the spring-like properties, in this case, particularly of the tendons, to store energy and return that energy during that hopping cycle. So that if you look at the work that the muscle needs to do is very low, whereas because it's all elastic storage in the tendon. So this is just one example how animals have learned to leverage mechanical properties to simplify their own behavior, in this particular case for energy storage. So what we wanted to know is, do humans leverage the impedance of their limbs to simplify neural controls in tasks when the mechanics of the limbs are beneficial? Okay. And so we looked at 3 different tasks. We looked at in one case where we looked at different tasks that required different torque motion profiles. And these are all going to be studies done at the ankle. The first task was a case where the stiffness or the torque increased with increases in length. So this would be like a positive stiffness, like a spring-like property of the joint. And then we looked at 2. Yeah.
There's a quick question from Elliot.
Have you looked at, or have people looked at the way that hopping frequency affects this? Because the stretch reflexes have their own intrinsic loop time, right? And then that interacts with adjustable mechanical impedance. I just wondered if people have looked at that. You've looked at that?
Yeah, we have not. We haven't done any work in hopping. That's a great question. There have been Arthur Prohaska has done, and Dave Bennett did some really nice studies looking at how different loop times can affect tremor, but I don't know if anyone has done it in more functional tasks like hopping. It might probably, but I don't know. Does
one phenomenon that's pretty interesting, if you're a rock climber, if you've ever tried it, and I have, and I've basically vibrated off a wall, and it's called treadle leg or Elvis leg or something like that, and that sort of what seems to happen is that you get stuck in a situation where your mechanical impedance basically matches the loop time, and it just amplifies itself until you pop off the wall, or if you're better, you figure out a way to get around it. I just popped off the wall.
Oh, that's really interesting.
Yeah.
Nice, thanks. Okay, so the first task we looked at required spring-like properties of a joint, and then we looked at 2 other tasks that don't. So in one case, we asked the subject to simply maintain a constant torque as the ankle was moved through a range of motion. So exerting a constant force on your environment. And the other case, we asked the torque to decrease as the ankle length or the muscle length increased. So we have 1 task that's consistent with the way the nervous system can regulate mechanics, and 2 tasks that are inconsistent. Because we've talked a lot about at the beginning of the talk, how muscles are essentially spring-like. Oops. Okay, so this is quickly go through our paradigm. So we had subjects attached to a rotary motor in the laboratory that moved the ankle backward and forth through a range of, if I'm remembering correctly, I think it was 15 or 30 degrees. At the same time, the subjects were tasked with generating a particular torque profile using some visual feedback on a monitor there. And they performed 3 different tasks again. In all tasks, the ankle was moved sinusoidally, as shown here. First task, the subjects had to generate a torque that was in phase with the motion. So this is the spring-like result. So the torque increases as the angle increases. Okay. This is the task that's consistent with limb mechanics. And again, if you plot this as a change in angle versus change in torque, you get a positive spring-like result here. The second task asks the subject to create a torque that was out of phase with the motion. So this requires essentially creating a negative spring, something that's exactly the opposite of the intrinsic mechanical properties of the ankle. And finally, they were asked to maintain a constant torque, which is essentially behave like a spring that has 0 stiffness. As they were doing this, so then we've come up with a technique that allows us to separately assess the contributions of the mechanical properties of the joint and the changes in neural control to the net torque about the ankle. There are essentially 2 steps. First, we have a way to measure the impedance of the ankle throughout the entire range of motions. This is a time-varying system identification technique where we essentially apply small random perturbations, we measure the torques, and we estimate the mechanics at each point in this cycle. And then from that model, we can then put in this sinusoidal movement and predict the torque due to impedance, or the torque due to the mechanical properties of the ankle. The second component is estimating the torque due to changes in muscle activation. So we ran a separate experiment where we had subjects generate sinusoidal torques in the same posture. We measured the electromyograms from the muscles acting about the ankle. We simply summed them or averaged them together into net EMG, and we found that that net EMG was correlated very well with torque. We just threw a square root, a parabola-like model here. So this is a simple static model relating EMG and torque, and it works extremely well under these static conditions, and I'll show you it works pretty well under the dynamic conditions as well. So now we have a way to predict the torque due to muscle activation, the torque due to impedance, and we sum these together to get the net torque. Okay, so we have our 2 components, torque generated by the nervous systems, which is our measure of how complicated of a task this is, and the torque that just arises from the current mechanical properties of the ankle. We found, first of all, that the errors in our torque predictions were small. So this simple model we're using that has these 2 components, the impedance component and the muscle activation component, predicted torque consistently well across all of our tasks. So we have our plus K task where these are the tasks where the mechanics of the task are consistent with the mechanics of the ankle, the 0 K task and negative stiffness task, and the errors were about the same in all of these. Now let's look at precisely what these models predicted. So what we found is, first of all, that the muscle activation torque, so the job that the nervous system has to do, changed dramatically across these 3 different tasks. So on the top here, again, you see the motion that the ankle was moved through, and we're plotting this in a phasor plot on the right here. Here's the torque generated by the subject in one of these tasks. This is the task where this motion torque profile was a positive stiffness consistent with ankle mechanics. And you can see that the subject was able to complete this torque pretty well. But let's look at the components of this torque. First, you can look at the component due to impedance. The impedance is largely in phase with the motion, and you can see that here. And you can guess what the nervous system is going to have to do is it's going to have to increase torque a little bit in the direction of the target here. And indeed, that's what we found. So I'm showing you results, these are average results from all the subjects in our group, where the impedance was aligned in the direction towards the target, and the nervous system created a little more torque in the same direction to get the net torque needed to complete the task. Now in the case where the subject was asked to maintain a constant torque, again, the impedance was still in phase with the motion, as can be seen in the polar plot here. And the nervous system, in order to bring the target back to 0, had to generate a torque in the opposite direction. Okay. And finally, for the negative stiffness task, again, the impedance was at about 0 degrees here in phase with the motion. The nervous system had to generate a large torque in the opposite direction to compensate for the mechanics of the ankle. So you had the mechanics of the ankle helping one of these tasks, and the nervous system could either leverage those mechanics or had to compensate for those mechanics. So if we look at the muscle activation torques and across the 3 different tasks, we can see that both the mean torque and the standard deviation of the torque was lowest in the task where the mechanical properties of the ankle were aligned with what the subject was asked to do. So the job of the nervous system in this case was much easier. In contrast, if you look at the impedance torques, too many screens open here. If you look at the torque due to impedance, it's the exact opposite. So it's largest in the case where the impedance of the ankle was aligned with the task demands. Now, what I think was interesting was we also asked subjects which task was the easiest. So we simply did an assessment of their perceived difficulty. And across all the subjects we tested, most found that this task where the impedance of the ankle was aligned with the task to be easiest. This was the case where they had to exert fewer changes in muscle activation. The job of the nervous system was simplest in terms of the amount of muscle torque that had to be generated. And this was also a task perceived to be easier.
So I'm wondering, so Alea Ahmed takes oxygen metabolic measures in very simple reaching movements. And very surprisingly, I find she actually, with different I would say subtly different mechanical conditions, she gets differences in oxygen consumption. So I'm wondering whether you're contrasting conditions where you actually create different muscle forces, torques, whether your question, what is easier, could be complemented with oxygen measures.
Yeah, that's a good question. I expect it would be because I would expect that the oxygen consumption would go up in the tasks where the mechanical properties were not of the ankle, were counter to what the subject was asked to do, counter to the behavioral demands. But let me just go back a slide. So I would predict that the metabolic consumption would be highest in the case where in this negative K test. Let me go back even one further. So if we look, this is a good slide to summarize it. So the task, if you look at the torque due to muscle activation, this is essentially of how active the nervous system was or how hard the drive to the muscles was. The task where had the greatest muscle activation was this negative K task. And I would guess that would probably correlate with metabolic demand. Mm-hmm. So that's an interesting thing to test. It doesn't correlate with perceived difficulty. So the task that was perceived to be most difficult was the one where I would predict metabolic demand to be in the middle, which is when they had to maintain a constant torque. But that's a testable hypothesis that would be.
But even within your tasks, that gradation of impedance, does that translate into different oxygen consumption, for example?
I don't think we can measure that, right? So because the oxygen consumption measurements are going to be very slow compared to these time-dependent changes in impedance. There's a couple of groups that are trying to infer, moment by moment changes in metabolic rate. But it's hard to prove if those estimates, hard to validate those estimates.
But as always, you just crank up the repetitions. Yeah. Matt has a question.
Yep, Matt.
Hey, thanks. Yeah, sorry if I didn't catch this, but how much training did they do? Like the curves that you're showing us, are those really like 0 through 6 seconds, or is that much later after some learning?
Yeah, so typically subjects would come in and train one day and do the experiments in another day. So so the tasks in the amount of training they required, we trained them equally in all tasks, but the amount of the tasks they found most difficult matches with this level of perceived difficulty. This maintaining a constant torque while this motor is moving your ankle back and forth was very hard for quite a few subjects.
Did you ever analyze the training data?
We have not yet, no. No.
Sorry, can I ask a question?
Sure.
I'm Mustafa and Dagmar invited me to this talk. I'm just wondering that you identified or estimated the stiffness of the ankle. And is it just related to the passive part? Because I saw that you added like the muscle activity to that. So this means that you identify the impedance just based on the passive part of the joint dynamics or mechanics?
No, good question, because it's a little confusing. So if I go back. Here. So this kind of shows our whole scheme for estimating the different components of the ankle torque. So what you'll see is that the, I don't have a time varying estimate. This is a hard one to see, but the impedance of the ankle changes as a function of time, or in this case, as a function of the sinusoidal movement. And so those changes are due to a number of things. They're due to all the things I mentioned in the first part of my talk. They're due to changes in muscle activation, and they're due to changes in joint angle. And of course, changes in muscle properties. So you can also, for any given impedance, you can change force. If you imagine, for example, I'm pushing against my cup here, right? So I can exert a force on this cup that's due to the changes in muscle activation. And so changes in muscle activation has two effects. It creates a force and it changes the impedance. And so if we look at the ankle torque here, what we assumed is that there's an impedance dependent torque that's due to externally, so let me go back to this example. If I exert forces on my cup, there's a change, I'm creating a force due to muscle activation, but I'm changing the impedance, but the impedance is not contributing directly to that change in force until I now try to move the cup. If I displace the cup, there's a change in force due to the change in displacement that's directly related to the mechanical properties. Right? So those are the two components we're trying to disentangle.
Exactly, because I was thinking that if it is just related to the position, it will be more related to the passive part. But for example, just consider that I'm activating both agonistic and antagonistic muscles. Without having any motion, I can increase the impedance of this joint. You know, that in this way, the impedance is related to the active part if agonistic and antagonistic are working together. So that's my question. That do you need to have muscle activation to estimate impedance as well, or just the position of joint is enough?
Yeah, we don't know. So we directly measure, the impedance of the joint by, so the top trace you can see here, this ankle angle. So this is what our experiment actually looks like. It's a sinusoidal motion, and on top of that sinusoidal motion, we apply a small perturbation. And we measure impedance as a change in torque resulting from that small change in displacement. Okay?
Yes.
So we have a direct measure of the impedance that doesn't look at muscle activity. It just measures it directly. Yeah,
but I'm thinking in like some people consider the impedance variation based on, like as I said, the summation or like the difference of agonistic and antagonistic activation levels. Like this is more active impedance or impedance change by, like this is intentional impedance. We can call this one like the passive impedance, like.
No, it's not passive, it's active. So those changes, the change in muscle activity. So in this particular paradigm, there was an agonist, there's always agonist activity because we had a bias torque that I didn't mention before. But in any case, there's a change in impedance due to changes in muscle activation as the subject does each of these tasks. And we measure that change in impedance directly by looking at the response to these small perturbations. So impedance is changing throughout time. It's not passive, it's definitely active.
Okay, thank you.
And then we estimate the torque due to that impedance by saying how much of a torque would you get by imposing the sinusoidal motion.
Yeah, I get that.
Okay.
Thank you.
Good question. Okay. So I'm going to skip a couple of quick things to be respectful of your time. Let me just say I've been a little sloppy in talking about impedance sometimes and stiffness other times. And the reason why I mentioned before is because in many of these tasks, stiffness is the component of impedance that dominates. And I just wanted to show you some data suggesting that. So this is a result from a single subject where we can take our measure of impedance and calculate the component of torque due to stiffness, viscosity, and the inertial properties. And you can see here the stiffness is shown in yellow, and that dominates the total torque over the course of the sinusoidal motion. And if we look at the standard deviation across all subjects and all the tasks we did, you can see that stiffness is by far, in all three tasks, the dominant component, which is why I've been focusing on that. So I think we've been able to show that if we create a task where the mechanical properties of the limb are beneficial, it simplifies what the nervous system has to do. Does this happen in tasks we do every day? I can't answer that question yet. But what's interesting to note is if you go look at a number of moment angle curves of behavioral conditions, this is a series of studies done out of Aaron Dollar's group, Chimay was the first author, looking at the ankle, the knee, oops, I keep pointing to the wrong screen, sorry, the ankle, the nip, and the ankle, the knee, and the hip. And if you look at these motion angle curves and just look at slopes, you'll see many portions of the locomotor cycle that have positive spring-like characteristics or behavioral requirements, suggesting that the spring-like properties of the joint would be beneficial in these cases. But it's not every task. So how do we perform tasks that are inconsistent with the limb mechanics? And I'm going to do this quickly, but the short answer is one way is through some of these involuntary feedback mechanisms that I mentioned earlier. So a couple of years ago, we completed a study where we had subjects complete a very simple task, which was maintain a constant torque. This was an upper limb study, but maintain a constant torque while being perturbed. Very similar to the ankle study that I just showed, the constant torque task there. And we also compared that ability to maintain a constant torque to the mechanical properties of the joint when the subject was asked just ignore the perturbation being applied to you. So in one case, they're trying to keep torque constant while perturbed. In the other case, they're distracted and not paying any attention to the perturbation. And we used, again, our results. These are the torques over the course of an experimental trial. We use our torques and our displacements to estimate the impedance, in this case, of the elbow. And what you can see is that in the torque control task, so again, this is as a function of frequency, the gain or the impedance of the joint. The low frequency component we can think of as our stiffness again. You can see that when subject was asked to control the torque, the ideal way to do this would be to have zero stiffness. For any time that there's a change in your joint angle, there's no change in torque. We can't do that exactly, but the subjects are able to lower the stiffness quite dramatically compared to this do not intervene condition. The question is how? How do they do it? So what we did is we started to look at muscle activity. What's the change in muscle activity in response to each of these small perturbations we're using to estimate the impedance of the joint? And what we found is that, so this is just a quick, this is an impulse response function where you're seeing the change in muscle activity under the two conditions. Take a look at the time scale here. So the change in activity and a negative sign is actually an increase in muscle activity. This is just a sign convention of our laboratory. If I were looking at a perturbation in the opposite direction, this would be positive. But you can see a big increase in activity at a relatively short latency. This is about 20 to 30 milliseconds in both tasks. And so what you're seeing here is a change in muscle activity from stretching the muscle. This is the short latency spinal stretch reflex. What happens after is where the interesting part happens. So if you look at the do not intervene condition, you have a steady state change in muscle activation. You stretch the muscle, you essentially get a tonic stretch reflex. Okay. In the case where the subjects are asked to reduce torque, what you'll see is they reverse muscle activity. And this reversal happens quite rapidly. It begins to happen at about 70 or so milliseconds, about where these longer latency cortical reflexes happen. So we haven't confirmed that these are long latency reflex responses, but these data suggest that there's a rapid change in muscle activity that happens before voluntary activity can occur that seems to decrease the stiffness in this task. And if we look at the change in muscle activity from those impulse response functions to the change in stiffness across all of our subjects, they correlate extremely well. Okay, so suggesting again that these involuntary pathways are one way to compensate for limb mechanics when the limb mechanics aren't aligned with the task that's being performed. So that ends my talk. Just to summarize the key points I tried to make in the second component. We found that tasks that are consistent with the intrinsic mechanics of our limbs require less modulation of muscle activity. So we think that these are tasks that are easier for the nervous system to perform. And this agrees with the perceptions of the subjects. Subjects perceive these tasks as being much easier. And finally, in cases where subjects are required to do tasks that are not consistent with the intrinsic mechanics of the limbs, rapid feedback pathways seem to be one mechanism that can allow us to compensate for limb mechanics when needed. So I'd like to end there. These are most of the current members of my group and a few of our funding sources. Thank you again for your time. I really appreciate this late in the day on a Thursday.
Okay, very good. So I'll clap here. Thank you so much, Eric. And sure, we are, as usual, at the end of our time, but I do want to allow and invite for a few comments, maybe make sure like within 10 minutes we're done. It's strenuous with Zoom, but let me open for general questions or specific questions.
One question I have is you pointed out that people can change the stiffness of a tendon by varying the load placed on that tendon. Is
Yes.
there any other way they can change the stiffness of the tendon?
As a passive structure, no. So so not that I can think of right now. The one way you can do it, so it's really the same mechanism, but through co-contraction, you can, bias your stiffness so that the stiffness where a given torque could be different based through co-contraction.
Elliot.
Yeah, it's been a while since I got really familiar with this literature, but I was wondering what the understanding is now of task-specific influences on the short latency reflexes.
Yeah, good question. So there have been, the short latency reflexes can be modulated through a few things like the classic gymnastics maneuver, for example. But you can change the overall excitability. And there's a nice study out of Rob Carney's group showing that if you give subjects feedback of their reflex gain, so measuring this continuously, that they can modulate it. But in most functional tasks, people have, we've shown, and way back before us, Volker Dietz and others have shown that for the functional tasks, at least when you're interacting with different mechanical loads, the short latency reflex sensitivity stays relatively constant, and it's just longer latency reflexes that changes quite a bit more.
And how long does it take to train somebody to have, quote, volitional control over those short latency things?
That I don't know. I haven't done those studies.
Oh, okay. Thanks.
So the permanent changes happen. So there, Jonathan Wolpaw and colleagues have done a lot of studies where they change, permanently change, not change within a task, but permanently change the sensitivity of the short latency reflexes. And those happen over a span of months.
Okay, thank you.
John?
Thanks for the sort of cool, wide-ranging talk. I remember hearing in various contexts people talking a lot about the importance of muscle fascia in a lot of these kinds of conversations, and I don't recall you mentioning that. I was wondering if you could comment on that aspect of muscles.
Yeah, that's a good question, and the answer is complicated. So, which is a short way to say I don't know the answer, but it's a big debate in the literature right now, right? So everything I've been referring to as muscle stiffness or muscle fiber stiffness will absolutely include extracellular matrix, so and potentially fascial connections to other muscles. There's two camps right now, some that argue that the extracellular matrix contribute nothing to the mechanical properties, and others that can argue it contributes everything. And it's interesting, it depends on the model you're looking at. So there's some really nice data out of the frog and other amphibians showing that in those conditions, the extracellular matrix seems to bear very little load within a muscle, and it's more the actin myosin cross bridges as well as the titin connecting them to the rest of the muscle that contributes all the short range stiffness that I talked about. However, if you look at, start to look at mammalian muscles, then it's been shown that extracellular matrix is quite a bit more important. So it's somewhat species dependent. I'm guessing the extracellular, think that extracellular matrix is quite a bit more important in human muscles than it would be in frogs, but that's still an open question.
Yeah, I also recall hearing some interesting conversations about like the anisotropy of fascia, where it's very stretchy in some directions, very stiff in others, and how that can, affect things at different time scales and all sorts of crazy stuff going on with that.
Yeah, that's absolutely true. And some of that isn't really going to come into, I think that's really important in a lot of pathologies, less important in the kind of mechanical measurements I'm making here, except possibly at some of the whole limb measurements, they could be important.
Just thinking along those ways. Oh, sorry, Randy. Just you're next. So when you were to proceed your measurements with flexibility stretching training, which not only warms up the muscles, but I think it does something to that extracellular matrix, so that you take this as an intervention to assess the contribution.
Yeah, those have been shown. A few groups have looked at that before and shown that you can have changes in the stiffness of a joint or a limb, but it's not long lasting. So there have been a lot of long-term studies on stretching, and surprisingly, even though it feels really good, they don't have a measurable change in the mechanics, at least in the studies that I've seen.
Okay. Randy.
Hey, Eric, it was a great talk. So my interest is in these neural determinants of limb impedance control. Mm-hmm. And one of the things I'm curious about is how prepared a person is with the environment that they're about to interact with. And does that influence where within this nervous system, whether it's more spinally driven or supraspinally driven, pathways that may be contributing to this modulation of stiffness that we see under these task-specific conditions. Yeah. And I was curious to know what your thoughts are on these determinants, because I feel like it's a little fuzzy to me in the literature as to, these proprioceptive pathways and whether they're really just, even though they're involuntary, that they may be looping around in the cortex or even in the brainstem. So I thought maybe I would be interested.
Yeah, that's a good question. So we have a whole set of parallel studies that I didn't talk about. Traditionally, when people talk about, so now we're talking about longer latency reflex modulation that happens across different tasks. And traditionally, when people talk about long latency reflex modulation, they assume that there's a single mechanism. And I think we're quite, in all the tasks that I showed today, where you're looking at modulating reflexes to compensate for different stability, the stability requirements, we've been able to show that that relies on the primary motor cortex and feedback through the primary motor cortex. So that's a long loop cortical mechanism. There have been a lot of analogous tasks where subjects are asked where they're interacting with a predictable environment. So an unstable environment is by its nature unpredictable. You don't know if you're going to go right or left. If on the other hand, you're given a predictable environment, but the timing of the disturbance is unpredictable. For example, let's say I'm your younger brother and I come up and I'm going to push you, and you're going to have to react in a certain direction, but you don't know when I'm going to push you. Well, then you can prepare. So you can have a motor plan in place for that. And in those cases, in what happens if you look at the response to the stretch, there's still a change in the muscle activation based on the task you're doing that occurs at a very similar latency, 50 to 70 milliseconds, maybe a little bit later, but it's very close. Those responses when you can prepare a task seem to rely on subcortical structures. We've been debating this with a couple of the groups for years, but I think it's quite clear. And we've been able to show, and the way we know that is that we can ask subjects to prepare a motor task. We stretch them, we get a response. The long latency stretch reflex is what we think is subcortical. we can do the same thing. We ask them to prepare the same motor response, and then we don't stretch them. What we do is we give them a loud acoustic startle, which is known to activate brainstem pathways, and we get the same response, right? And so that's why we think at least the subcortical, I think what we've been able to show convincingly is that at least this can be released by subcortical pathways. We can't rule out cortical pathways. But so I think and I'm sure there are other mechanisms as well. Those are the two we've studied. And so the long answer to your short question was, if you can prepare a task or you're interacting with an environment where you can prepare a motor response, then I think subcortical pathways are quite important. If you can't prepare a motor response, it seems to be motor cortical.
All right, so James has patiently waited, but let's have James as the last question and then call it an evening.
Thank you for the presentation. My question was about the stiffness during motion. So you had mentioned a couple times throughout the presentation that it changes drastically during motion. I was wondering what your thoughts are on why. I guess maybe that was somewhat related to one of your later topics.
Yes. So why I can't answer. The one thing we have just recently answered is that we know where. So we've combined the study I showed you where we've been able to decouple the tendon component and the muscle component, which may include ECM component, and have been able to show that the tendon component decreases a little bit during motion, but is relatively constant, and the muscle component decreases dramatically. So it has something to do with, it could be cycling, moving these cycling cross bridges, or it could be interaction between the cross bridges and the ECM. So I don't understand the mechanism right now, but we at least know where the change is occurring.
I see. Thank you.
Yeah.
Okay, so I think our first virtual action club was a successful experiment. Thank you, Eric. So that was great.
Appreciate the great questions. Nice seeing good friends.
Yeah, and thank you all for chiming in and behaving as normal as is possible. So I think this was great. Under normal circumstances, we would go out for dinner now, have a glass of wine. So let's hope that this will return soon. So thank you again. We have the next action club is Aaron Batista in 3 weeks. So looking forward to it. And Eric, you can join and pester other people.
I think I will. All right. Thanks again. See you.
Yeah.
Bye.